roster up there. That's my thing there. So I'm going to roll real quick. I got almost all of you in here. And I'll turn off the other light here so it's centered. View from the present. And away we go. Whoops. One thing real quick. Before I start here, I wanted to go to here real quick. Right? Just so you see it, even though it'll be on the video. That's the biggest thing. So if you look here real quick, this is where we're at. Okay, so I added this in Invert PowerPoint Part 1 because it's a big one. So I have the introduction to the animal. So it says right there, please use this PowerPoint for your main study. This is the main hub one that I go over. And then this is the supporting one I talked about yesterday. This is a resource. Do not turn in. It's with the outline. If you wanted the outline, some of you picked it up yesterday. It's here. Feel free to do that. And then the assignments so far. Okay. So I just wanted to point that out. This is this PowerPoint we're starting here today. So like I put it right at the top of the unit. Okay. All right. So here now we can go here. View from present. All right. I'm back up. All right. Okay. So you should be able to see this. That's centered pretty good. If not, I'll try and keep my shadow out of here and point with my ruler. I know you will see my ugly mug creeping back in there. Okay, so guys and gals, one of the biggest things about the invert or the animal kingdom is, like anything, these first few slides is just talking about the general whole hub of the animal kingdom, okay? And I refer to the 7 system analysis, okay? And I'll explain those here. That's the first PowerPoints here. So I laid out this PowerPoint as best of an outline as I could. I tried to even make sure the subscript, underline key terms that a lot of you already know. You know what multicellular is, what heterotropic is, hopefully, the term invert. So I tried to also help you with vocab and going ahead and defining out those. If I throw in vocab terms sometime, which I will, that you're studying the same definition prior to the quizzes or exams or whatever that I'm instructing you on to. And I taught you guys before, staying off of when you Wikipedia a term or whatever, it may not be worded the same way in which I put on the test like I did for you guys at one time, okay? So what is a basic animal? So we're talking the term vertebrates and invertebrates, okay? And so for simple terms, when they're talking vertebrates and invertebrates, they're talking simple backbone, okay? So we are vertebrates, okay? Animals, amphibians, reptiles, mammals, okay? We are all vertebrates, or like you've learned in your Big 50 or your modified project, chordata, which means central notochord. We have a spinal cord that's infused and surrounded by bone, okay? Only 3%, and that stays true to fact, even though we're constantly finding new species, most of the new species that we're finding, okay, I wouldn't say all of them, but the majority of them are invertebrates because they're sea-bearing, or small or aquatic, bacteria-based or something. So in those non-animal kingdom type stuff, but when we're talking about the animal kingdom in itself, 3% of all organisms, give or take 0.2 or something like that, so I just rounded to 3, have a backbone, okay, or considered vertebrates. So that means the 97% of all the rest of the animal kingdom do not. And the biggest reason why is that, is you haven't learned already, arthropods, insects and spiders, and that there's so many of those species. So they make up, like I said, 97% of the animal kingdom. A funny fact is, if you were to go on to the rainforest in Brazil, okay, the Brazilian rainforest, the Amazon, okay, one of those giant trees that you ever see in some of those cool videos and so on can have as many as 200 different plant animal species on one tree. One tree can house as many as 10 different individual species of ants. You're like, really? I thought it was only like red ants, fire, this, that. But I mean, you got to remember, a lot of organisms, especially insects, are defined possibly also by their behavior. They may look almost exactly the same, but their territory or their characteristics or the behavior, so they make them as different species or subspecies. So then that adds to this 97%. But that's one of the cool things, is there's so many of them out there, okay? And when they reproduce on such a large number, and their genome or their genetic makeup is so basic, they are able to mutate or change or evolve, is the key word there with it. So you see a lot of diversity within that kingdom. You throw in a bunch of time, you have all these different little organisms that are really similar, but they're different. And as you've noticed in the classification books, like the TAN insect book, that's why I've explained to some of you, especially yesterday, with the species, they don't get as specific because there's so many of them. And just in the last decade and a half, they're really breaking down those into these little fine subgenuses and subspecies and that stuff, okay? All right, so, and they're the most diverse of the all kingdom just simply because of the basic characteristics that they serve, but mostly is because of the insects. Okay, so here's some characteristics, and I sub put them in there as A, B, C, D, and so on. So one of the main things, a characteristic of the animal kingdom, is they are heterotropic, okay? Heterotropic basically means we have to get energy from the nutrients that we take into our body, okay? So we're not phototropic, we're not autotropic. I mean, we don't take light and convert it to energy. We don't, take the sun like the chloroplasts and so on like a plant. We have to eat, okay? We have to feed on organic, carbon-based organisms, right, to make up our energy, okay? So that's one big common characteristic. We are definitely multicellular. Not like a bacteria, a one single-celled organism. We have multiple cells that are all combined together to form tissues, and those tissues make up organs and make up the systems and so on as well. We are eukaryotic, which basically means we have a nucleus, so a hub of the nucleus with membranes around, and then outside that nucleus, as we know, a little review, we have lots and lots of organelles, just like we have our liver and lungs and kidneys and intestines. Well, in some cases, the term organelles means tiny organs in Latin. So all those little things found outside the nucleus, which you think are not important, they're extremely important, because those things right now on millions of levels over in your body, you as a human, okay, are doing a lot of very important functions as a whole together to maintain listening to my voice, or like we use the term homeostasis. Some of you are my AP students. Homeostasis means to maintain life functions, blood pressure, body temperature, simple awareness stuff, okay? Okay, and then we also lack a cell wall down here in the right corner here, D. So A, B, C, and D, they lack cell wall, all right? And the last but not least simple characteristic, I only had it clear up here on the page. Sorry, it's all the way at the top, but you'll be able to see it in your PowerPoint. We what we call a division of labor, okay? So, it's way up there at the top. So what this says is bodies contain specialized cells that cause different tasks of labor. So what that is saying is lung cells are lung cells, liver cells are liver cells, skin cells. So each cell, even though it has the same basic function, which most of you, if not all of you, can cipher this out without me explaining it, they all are the same basic program, like an assembly line, okay? And when cells come down that assembly line, okay, they are then programmed through a thing called cellular or protein synthesis, okay? And I know some of you guys remember that from the little A, T, C, and G in your days of biology, where the little polypeptide chains, and hopefully some of the words that I'm throwing at you, like, oh, I remember that, I hated that unit, okay? But basically what they're doing is forming these coded systems that then attach these amino acids, like proline and aniline and tryptamine, and these ones, there's 20 essential ones for us as humans, 20 essential amino acids that make up us. And when they combine, think of it like an alphabet, the combination of these shapes and puzzles, and it's like a lock and key, then put in motion for those particular cells to do their job, i.e. a nerve cell, a skin cell, a liver cell, so that's why, transplanting just any old generic cell is so important, which brings me to an important task. If you've ever heard of the term stem cell, okay, that was a very, very important topic, I want to say at least three elections, I could be off, but 3 elections ago or so, about stem cell research, and a lot of people were misinformed about what a stem cell is. So I always would tell my students during the cell unit is to understand a stem cell. When you hear that term, the stem cell is a cell that has not been given a job or division of labor. It's basically like the assembly line at the Ford plant where you got all these F-150 trucks coming down and you see the same body frame and the doors and the rearview mirrors. But as they get closer to the end of the assembly, they start adding in leather, or they add in this wheel or that, or some type of structure of that nature that makes it specific, like a Laramie or a 4x4 or a sport package. You get my picture. So a stem cell, when they were talking about that stuff, when they were talking about fetal stem cells after a newborn was born, after childbirth, okay, awesome like that, the placenta, which is the afterbirth, which is attached inside mom's uterine lining, detaches with the umbilical cord, snipping of the umbilical cord, baby's doing great, this and that. The afterbirth comes out. Inside that umbilical cord is obviously a lot of blood, okay, and stuff like that. So this is considered biohazardous waste. It's discarded. It has nothing to do with the child. So what they would do with stem cells, they would harvest the stem cells from the umbilical cord that were going to be wasted anyway because those cells in that umbilical cord were not programmed yet. So then they would process, and I'm summarizing really quick, but then they would take these cells, put them in a nice safe place, that then in a lab, say, example, they could take those cells and manipulate them to possibly turn them into skin cells or liver cells or something like that. Say in a burn unit where they have to recombine new skin cells for somebody that had burns for skin grafts. They could literally grow artificial skin, which is from real cells, but they program them in a petri dish or in a lab or something like that to help somebody that's been injured. Or I remember in one particular case, a rare genetic disorder with this. I was at a science convention, it's been years ago, and the young man would be in his 30s now, have to be, but he had a rare genetic condition in which his bladder, little tiny bladder, okay, transition tissue, when it fills up with urine and waste, your bladder is the only place where the tissue expands, and then it goes back to its original size after you go to the bathroom. Well, he had a rare genetic disorder where the tissue started to break down, so these like little pinholes were going to form, and his bladder would leak. And as you know, that's urea and urine and salts and various impurities that we get out of our body. What was happening was it was like leaking like a dripping pipe or a faucet inside his body cavity, and was causing infection or septic. It could kill him. So, long story short, taking stem cells, and they abstracted actual skin sample from his bladder. They coded it out. They saw the sequence. They then take these new cells that were non-programmed, this is true, and they programmed them and basically programmed those non-cells to be this little boy's bladder cells. They grew them in a quicker thing in a petri dish around this little orb, which was kind of like a plastic orb, the same size of his bladder. We're talking a little boy here under the age of 5, so this bladder, when it's just normal, is not any bigger than maybe like a lemon, when it's not full with urine, okay? So they were able to grow this synthetic thing in a faster pace. Then they did a surgery. They replaced the grown one in the lab, and his body didn't reject it because it was his own cells. And they had these extra cells that they were able to program because they were able to code it from one of his healthy bladder cells, do it outside a lab from the stem cells that they had abstracted, and then put this back in this little boy. And he's grown full life. And when you explain like that, people go, oh, so that's what stem cells are. Oh yeah, I think that's a great idea. So it used to be a lot of times the misconception that they thought, young children or young, things were being hurt. No, they were, so now when you talk about adult stem cells, that's a procedure like myself. If I wanted to donate my adult stem cells, that's my choice. I'd sign a consent, and it's very painful because they abstract those from like the marrow in your hip or your femur or something like that. That's a completely different thing because you're a consenting adult, and if that's something you want to do for the better good of science, that's okay, because stem cells are unprogrammed cells in that. So, thanks for being patient on that, but that kind of gives you an idea of that story, and it's a real one. That's just one of many now that are off the chart about what they can do with this stuff now. But that's how, in a nutshell, right now, cells that are being reproduced to replace old, broken down, or wore out, or just adding to because you guys are young, they're being told what to do. They're having a program, or a barcode, or a game plan. Okay. So in a nutshell, that is also a main thing that eukaryotic cells do. Okay. So here's the 7 essential things I talked about that in each phylum or each class. And today, Periphery, it'll go fast. It's on like 5 slides after I get through these other 5 slides. That very obvious. Okay. So for example, feeding is a very important structural function of obviously. So there's some I left out, but basically I tried to list out all the ways in which animals feed. There's herbivores, obviously, which eat plants. Carnivores, that's us. I left off the term omnivore, which is what we are if we eat plant and animal. Omnivore eats both. So I forgot that one on there. Parasites, which are parasitic, live off and feed off the outside of a host. A definition of parasite, too, is parasites' intentions are not to kill the host. Sometimes they do because of their presence, and but parasites need, like a mosquito is a parasitic organism that feeds off the body fluids, a tick, a leech, all right, things of that nature. Those are considered parasitic organisms, okay? Filter feeders, things that strain out, camera, don't go off on me. Filter feeders that strain tiny organisms from the water, from whatever purposes. You will see a lot of filter feeding with some of these early invertebrates because they don't have any advanced feeding systems. Detritus, or dead and decomposition. So this is just the fancy word for the dead and decaying material. They feed on bits of decaying plants and animal, funguses, and from the fungi kingdom, and various other things that feed on this stuff. A lot of times they're microscopic or very small organisms that feed on that. But the carnage, we watch a lot of our TV shows. There's a lot of animals, a lot of rodents, a lot of smaller carnivora type things. Your wolverines, your skunks, a lot of those organisms, even fox and stuff. A lot of their feed is cleaning up, I guess, in the forest, kind of like organisms that have either died of natural causes or from this or that, but they're feeding on these different materials. All right, respiration is the second one. Okay, so breathing, obviously, must be able to consume oxygen. That's the thing. So basically, also to oxygen and CO2 are what they call blood gases. Okay, because they're the gases found in our blood. O2, CO2. Okay, small animals, you'll see this a lot too. They diffuse. Remember, diffuse is going from high to low concentration across the membrane. Gases through the skin. Amphibians are big with that because of moist skin when we get with that. And there's a lot of other inverts that use diffusion, and you'll see that before we get more advanced. Large animals need specialized respiratory cells as they get older too as well, like ourselves, mammals, reptiles, and so on. Especially when we started becoming land-based and we had endoskeletons and we started changing body structures so on to support and not have the ability thing for our lungs to be able to inflate, like the development of a diaphragm, which is a strong, oval-shaped muscle that helps inhalate and exhalate our lungs. Okay. Okay, so there's respiration. Okay, number 3 is internal transport. Okay, so you got internal transport. Some small aquatic animals function without internal transport. So basically what it's talking about is like our bloodstream would be our internal transport. Some organisms don't need it, and you'll see that especially in the early phylums that we talk about. And you'll see a slow transition. The more advanced they get, the more specific or ways of getting things where they need to go to specific tissues because of division of labor. You will start to see that develop. Okay. Large animals like ourselves, we have a much evolved circulatory system in which, like I said, for example, our heart, blood vessels, and so on. So those are things that you will see as we neck our way up from very basic to more advanced. We will see that stuff start to show up. Okay. Excretion. Oh yes, the ever more popular getting way of liquid and solid waste. Very, very important. Nitrogenous waste, it's called a lot of times. All right. Toxic materials that need to be filtered out. For example, your kidney right now is detoxifying impurities in your blood system. Okay, they use a term called shunting. The blood is shunted through your liver, which neutralizes a lot of the toxins. At which time, then that blood is being filtered through your kidneys, through the your nephrons. Okay, tubule type stuff, kind of like a oil filter, taking out all that, cleaning out your blood. All right. So then it goes back in, oxygenate, and does the whole cycle and so on. It's a necessary factor. Okay? If something was not working properly with your kidneys, you might know somebody or some individual that's had something where they have to go through a condition called dialysis. Dialysis is where individuals have to go in and actually hooked up to an artificial kidney that every drop of their blood is pumped out of through one, goes through an artificial machine, clean, and back in their body. It's a normal procedure. Because if your kidneys are not able to do that, I like to think of it like sludge, but the stuff just keeps getting thicker and thicker. And think of your blood not being nice and thin and easy to move. It starts getting thicker like syrup and oil and so on. And as you know, the medical stuff and so it goes with that. So it's very important with that. So you'll see the development of cellular metabolism, how it diffuses, and how we get actual structures that show up to do that for us. Okay? 3 down, 4 to go. Yep, I said large animals that develop excretory systems and so on. Okay, so that's 4 of them. Okay, all right. And 5. Okay, response is our central nervous system. So that I need to put that a little closer down. I'm gonna turn that camera down just a smidge here. Let's look at that top. Doing good. There we go. Okay. So response would be, as we AKA know it. Bam, I'm back. Response, animals, nervous system, okay? Stimuli. If I was to go, and scream real bad and somebody have sleep and you startle, Myra, why did you do that? I just stimulated your central nervous system, and I startled you, I didn't scare you, and you might all of a sudden have that little adrenaline dump where your feet tingle and so on, and you're like, okay, I'm awake now, okay? That would be in a response to an environment. Or if you're, say, walking down the sidewalk and you hear cars going on the side, down the road, you process that, no, those are cars going by, I'm on the sidewalk, they're on the road. I process that as, I know what it is, it's a non-threatening noise, but all of a sudden somebody who knows you thinks it'd be funny as they get up beside you, they lay on their horn. You don't go and jump into traffic, you pull into here and you flinch, maybe you yell something back at them, but they startled you. Because your reaction is, your fight or flight reaction would be your central nervous is reacting to that potential possible threat, okay? Or if somebody you've seen raises their hand and you flinch inadvertently, or you get the picture of our central nervous system with that, okay? So we identify our own kind. Animals use specialized cells called nerve cells. You'll see a massive development of that. The term stimuli is anything that generates a response or we have to do. You'll start to see organisms that develop actual brain and nervous systems, which we are familiar with, okay? And determine how the animals respond, okay? Movement, pretty simple. Some animals, there's a term that you see right off the bat here with sponges in a second, meaning sessile, they don't move, they stay anchored. Most organisms in all groups remain with the term that they call motile. And I don't have that one here, but motile, you'll see that with the periphery, means they mobile, move. Most animals use tissues called muscles, obviously. And like I said, this stuff is very elementary or basic, but we don't ever think about what defines an organism. And it's seven basic systems, the essential life functions. And this is number 6, movement, okay? All right, number 7, next last one, reproduction, okay? You will see us a lot of times talk about asexual and sexual reproduction, okay? You will see a lot of organisms, especially in the invertebrate that do not have defined sexes, or they can go literally back and forth depending on the environment. There are certain organisms that, depending on the environment, they can switch male or female, or they have both functioning, what they've heard of the term as hermaphroditic. I think I might have that one. Yeah, no, not in this, but you'll see the term hermaphroditic means they have both functioning male and female reproductive systems, so they can, like I said, reproduce asexually and continue their species or organism moving forward, okay? So some, like for example, jellyfish, I use an example, they can switch back and forth between asexual and sexual reproduction, depending on the environment. Could be temperature, could be season, could be a lot of different things, okay? Some bear live young, as we know, eggs. Direct development is obviously baby animal looks just like that. Adult increase in size, that would be like our cells and mammals. There's a lot of indirect development, which would be more of your metamorphosis like in your insects and arthropods, okay? Right, where they go through stages, larval, pupa, things of that nature as well, okay? So there's just some different stuff there, and they change in shape. All right, so this one these are just, some general terms here. Trends in animal evolution, okay? For example, radial symmetry as opposed to bilateral symmetry, okay? These are terms you see a lot of times too as well. Levels of organizations become higher as they become more complex. I will point that out and you will see that. We're starting at the basic one here in a second, the phylum Porifera, okay? Simple animals can carry out essential functions at the cell, but here's the thing is, they may be simple functions, but they're getting the job done. And some of these organisms in these groups, for example, Porifera are the oldest group of animal organisms. They've been around for millions of years. And we know that because of fossils that we found, because of the radioactive dating, so they're as basic comes, all right? And their tissues, the more complex animals have, the more specialized cells, like our cells, we're as specialized as they come, right? There's 2 types of layout. There's what we call radial symmetry, like a starfish or a jellyfish, where a radial means from a body part and they radiate out from a central hub, like a pizza, okay, radial, okay? And they have a central point. They have no real head or tails. Starfish, sea anemones, are like sand dollars and things like that, are example, okay? You have your bilateral symmetry, okay, which is basically left side looks like right side. That'd be us and various other organisms as well. And then the terms, what they refer to as anterior, posterior, dorsal, and ventral. My anatomy and physiology, nursing students and so on, you've heard these terms left and right. Anterior is front, posterior. If you've ever been to a PT or an OT or a physical therapist, you might have heard them, the doctor checking your proximal and distal and anterior and all these numbers, and you have an assistant writing down the diagnosis. Maybe you had a rotator cuff or a knee surgery or something of that nature. And all these terms are associated basically, instead of them saying, well, the front of your shoulder or the back of your shoulder or things like that. Upper, lower, okay? Complex animals have what they call cephalization, okay? We develop as an organism cephalocaudal. The term cephalo refers to head. So everything centers around our head, where our brain is, our sensory receptors, cephalization, okay? Concentration of our sense organs and nerve cells are in the anterior end, or at the front, or at the head, okay? So that's an important term. You hear cephalization a lot in child development classes and in child psychology class, when they talk about that. So when you hear the term cephalo, it's referring to head, okay? So we're talking about cephalization, okay? Due to the fact that most animals move head first, that is why, okay, in that particular case. And then, of course, you will see this term, small clusters of nerve cells are called ganglia until they become more refined and defined until we call them a brain. But we go from simple nerve cells to a couple nerve cells, and then they call this ganglia because it's not as sophisticated as a brain. And then you will eventually see, as we go up through the hierarchy from your inverts into your vertebrates, they will start referring to things as having a defined brain. But it's their nervous system. Large numbers of nerve cells, and of course, called brain cell, right? But so these are basically, the ganglia is basically a brain, but 1 or 2 or 3 cells is not classified the same as ours. Okay. All right. So there's just a picture: bilateral symmetry, radial symmetry, showing you those pictures. Pictures obviously define that a lot of times. Very simple. Here's showing, anterior, posterior. Ventral would be belly. Dorsal, like dorsal fin, is the back. Posterior is the rear. Anterior is the front. Okay. In radial symmetry, oral is where the mouth is. Aboral would be considered like the back side or the underbelly side. So they use the terms oral and aboral. Oral referring to like the oral opening or the mouth or where they take things in. All right, in the home stretch, you guys doing good. Almost done, and then I'll let you guys go. We're in the first phylum, sponges, okay? All right, very simple stuff. So I put them out in simple terms like this. First of all, most live in the sea, not with SpongeBob, okay? But most live in the sea, but a few species do live in fresh water, okay? So you could go up in the Great Lakes and so on, but the majority of them are saltwater-based or marine-based organisms, okay? The term porifera means pore bearers. So obviously sponges have all the little pores. That's how they filter stuff through. So this is Latin, Greek for sponges, or that group of organisms that fall into that. They were once thought to be plants. Makes sense. There's a lot of different shapes, and at first when you see them, they look like underwater plants. But then as we researched them and understood them, we realized they don't have the same layout, as a plant. So therefore we knew within the animal kingdom, they didn't go in the kingdom Plantae, they went in the kingdom Animalia. Okay? Most biologists believe sponges evolved from single-celled ancestors separately from other multicellulars. And the simple fact was, think about this, and you're going to hear me refer to this a lot. Scientists believe nobody was there during the cool division of all these amazing organisms and facets and structures. So it's speculation, because we can only go on, like I told you, with radioactive dating and so on, the fossils we find, the evidence we can see, plugging it into the technology we have, and just, common sense or plugging in. So, a lot of these are speculation or hypothesizing or extremely well-educated guesses, because when you look at how they're laid out and you compare them, like, for example, to single-celled organisms such as bacteria, and there's nothing that when you get from bacteria, the next organisms that have a little difference, that go up a little better, are the sponges and how they're laid out, and they're considered to be multicellular, but they're not real advanced. So it's kind of one of those things, until we find something else, we're pretty sure we think the multicellular organisms, okay, came from obviously the basic single-celled organisms. And why does that make sense? Well, anything in general does not evolve backwards. We take a basic model like cars and cell phones and computers and video games and whatever, and the job is to improve or make them better. We don't recess and go backwards, because in the natural world, if we don't continue to move forward, okay, or we move backwards, you risk the chance of extinction or struggling with homeostasis or life functions, okay? So it makes sense, until we find something else in it, okay, that sponges, okay, fall right in there as how they're set up. And if you had to put them in a hierarchy, like a cladiogram, it makes sense, okay? All right. So, form and function. So we run through the system analysis with the sponges, and they go faster because they're so basic, guys and gals, that they don't have 7 distinct systems like I just showed you. And you'll see that with a lot of the earlier inverts. So for example, forms and function. Basically, the body of the sponge has a wall or an epidermis or an epidermal layer, okay? All right, and then they have a cavity. So it's the outside and the inside. There's not a lot in between, okay? But there is stuff in between to make them perform life functions because they are alive. So for example, the current, the water, what it means by current, flows in from obviously the outside through those little pores, okay? And as they're flowing through, they got these little flagella. That's a common term here. Flagella or cilia are just whip-like little tiny hairs that just basically undulate back and forth like this, or it'd be no different if me doing this, just taking my hands like this, I'm creating a breeze in towards my body. Well, they don't have a big fancy program, but the cells are called collar cells or conocytes, okay? Because they look like a collar. I mean, that's what a lot of times, these common names, they look like through a microscope. Wow, it looks like the collar of a horse collar. That's what it reminds you of. Have you ever seen those donut-looking things you put over the stock horses that the reins hook to? They call them horse collar, okay? So it looks like a collar, so that, hey, it looks like a collar cell. That's where the common name from, but in Latin means conocytes, okay? So basically, this specialized cell, its only job is to sit here all day and just make these little flagella and make this motion. Wave, wave, or undulate like this. Keep the flow of the water going from the outside into the inside cavity. Most of the time, the current is doing it for it, but what if I'm in a pool, or I'm in a pond, or I'm in an area where the water is very stagnant? I got to have these little collar cells keeping the flow of stuff coming through. Just no different than, even when I'm sleeping, I have my diaphragm muscle working, inhaling, inflating my lungs involuntarily, okay? That is a involuntary response. Muscles that continue to do this to carry out life functions because of my brain. So my brain sends that to that, just like it has all impact in one, these simple specialized cells that just basically, I don't know why I'm doing this, I just keep making these flagella, do this all day, that's my job. Simple, basic, but it works. So the water exits through a large hole, they call it the osculum, which is just basically in the tube. So then as the current flows through, it's bringing in obviously oxygen from the water, any small debris that's flowing through and so on. And then it carries away the waste as well. So as it's going through this little canal, it's bringing in oxygen. As it's passing out, CO2 like we're breathing out, but it's able, this collar cell is expelling CO2 that's already been processed. It's picking up food, it's picking up oxygen and giving out waste. So it's an all-in-one spot. So basically, this one little cell is performing all of the, literally almost all of the life functions, okay? Waste, respiration, circulation, so what, 4 or 5 of them, it's not moving, but it even has 6 of the 7 things are all centered around this, even reproduction, okay? So water also transports the sperm or the gametes. All right, here's a term you'll see a lot. The term gametes is Latin or Greek for sex cells. Gametes, i.e. example, are sperm and egg, male and female reproductive gametes, okay? So when you see the word gametes, or they refer to it, they say sperm and egg, or they just say the sex gametes, it's all the same. It's not something different. So you're going to hear that term a lot in these units, and you hear it a lot when we get into genetics later on second semester, okay? So don't let this see gametes in this. It's all the same thing, okay? Larvae are just obviously like little smaller versions of the organism being developed, okay? Many sponges have then these thin spines called spicules, okay? That basically look like little glass rods, are the earliest prehistoric form of what an organism would have to be what we call a skeleton, okay? They got these little glass silicate-like little rods, almost like, looks like little rock candy, that having these things present like little pixie sticks helps give shape and some strength and resistance to the outside structure. Otherwise, it'd just be like a slime blob falling all over the rocks. Helps give it shape. So this is the forming of these spicules, help give it the shape and form so we can tell what it is, all right, and give it some space, all right. So like I said, the spicules are made of like a silicate or a silicone type structure, natural element. The softer ones, natural ones, like the loofahs and stuff you can buy, not the synthetic ones you buy, at the store, but the real life sponges They have a stuff called spongin in them. That's the nice soft ones you use in the bath to exfoliate, you know, your skin and so on, to scrub your feet, hands, and body, and so on. Okay. Some sponges have skeletons of both that, you know, just depends. So they have the soft ones, they have the wriggly ones. Not all sponges is something you'd want to use for that structure. Okay. All right. The only other specialized cell they have, so they have the conocytes, and they have these things called ambiocytes. Okay, or I'm sorry, amoebocytes. Anybody ever heard of the term amoeba? It's that little blob-like, little water-based single-celled organism. So that's what it kind of looks like. But these things kind of inside the tissue, they basically help build the little spicules, the little glass rod type structures. They wander around the sponge and they do all the functions. They're kind of like the caretaker. They take care of getting food to where it needs to do to all the various cells, nutrients, gametes, removal and introduction of oxygen. So this one little cell with this little buddy, the conocyte collar cell, bringing stuff to them. There's the person on the loading dock bringing all the stuff to the loading dock, where these little ambocytes then take it and they distribute it all throughout the tissues inside the sponge. Okay? Feeding, they're filter feeders. Easy one there. Filter feeder sponges are filter feeders that sift microscopic particles. Okay? They don't have any teeth, don't have any chew, so as it comes in, it's absorbed. Okay? So like I said, as the conocytes draw the water and the tiny food particles in, they stick to the collar cells, okay? And the flagella, at that point they become trapped. Then the amoebocytes, they come along, they pass the food on to them, like I said. So the collar cells are like the loading dock guys that drop it off. The amoebocytes pick it up, take it into the store, use it what they need, okay? And all the parts of the cell. So then you see respiration, excretion, and external transport, those three, all use a process called, I have it in here, diffusion. No, okay. So I think it's on the next slide. So all three of these, because of the water flowing through, are all taken care of. All right. So remember, it's such a basic organism that they do not need specific structures. They just need the water to flow through, and it's taken care of, and the water to flow out. That's it. Okay. So sponge cells remove oxygen and give off CO2, just like we do by breathing. They do it a little simpler. Metabolic waste, which would be like urea waste, the stuff that, you know, the toxins that we take right out of all of our cells. Ammonia, very toxic. Our liver, which I'll get to that way down thing, our liver neutralizes the ammonia in our blood right now, okay? All right? So that's why we don't have a yellowish tint to our skin, or an orangish jaundice, if you ever heard that word, or a yellowish tint to our eyes, because the toxicity in our bodies is being neutralized from our awesome liver, and that's what it's there for. And then eventually, once the ammonia is sifted out, it's just carried out directly. Okay, so there's not a neutralizer, because they don't need something that fancy like we do. Okay, and last but not least is reproduction. Okay, so how they reproduce? They do have sexual reproduction. Okay, so basically, if you've ever seen, and I think in the video if you watched yesterday or on Monday, you saw the big tube things like jettison. It looks like a bunch of just material, like a fog out. That was, and they probably told you, that was their way of reproducing, is they blast all of their gametes out into the water, okay, to the coral reef, and then they let the current, and they settle in other sponges, so they reproduce that way. So the sperm gets released out into the water carries it, kind of like the same way pollen carries flowers, you know, and bees and things of that nature. The sperm, or the gametes, the male gamete then lands into another sponge. The ambiocytes then picks it up through the collar cells and so on. You get this little tiny larvae, look like little tiny seahorses. These little tiny little cute little things under a microscope. Right? And they swim around, they anchor themselves long distances for whatever reasons. You know, they go out and do their thing. And they also reproduce asexually as well. Freshwater sponges produce sphere-shaped things called gymnals. They look like little candy-coated pieces of candy. And these gymnals have this hard, tough outer coating that they're able to lay dormant when the water's really cold, okay? And when the conditions become perfect, kind of like how seeds right now will lay non-germinating, and they'll just sit there and wait in the soil, or wait till the spring when the weather gets warmer. Well, nature has a great way of keeping that intact in various areas where it's not tropical, where it's warm all the time. Freshwater ones, hey, that's a special adaptation because they go through cycles of the season. So when it's ample and it's necessary and everything's right, then it can germinate or blossom into that structure. All right, so it's basically got this little insulated like a little seed. Okay, they call them gymnals. Okay, all right. They can also reproduce by budding. Basically, the term budding is if you see that like a structure forms at the top, almost like the top of a lollipop, and then they just pinch off, and then they settle, attached to some coral or a boat or a rock or sand, and they can grow from there. Okay, so budding is just where part of the sponge simply just falls off of the parent and can grow a new sponge. So I like a lot of times in science you're going to see the term defines what it's doing. So budding is really it's budding, a piece of it's budding off, and it just attaches somewhere else. Okay, and then they have great powers of regeneration. Okay, one of the things I didn't add in here, there's actually extensive research with a lot of basic inverts about how they're so great at regenerating lost body parts or tissues. And they're using that technology to some level, synthetically producing that to help with, reproduction of organs, reproduction of material that, and so on as well. Okay? All right? So last but not least, okay, is the little slide here where it says how sponges fit into the world. There's some little extra tidbits there. This will talk about treating infections and diseases as well. Okay? Bathing, washing, and so on like that. Okay? All right. So, and there's the picture from like the diagram you'll be doing. There's a tube. You'll use this for today. Help you out a little bit with that, with the structure of the coloring guide one that I have. Okay? So no big deal. Okay? All right. So you made it. Like I said, you're almost done, and then you will not have to come back. So stay with me 3 or 4 more minutes, and I'll be done with you. Okay. So there's the slide. There's a collar cell blown up. Somebody's knocking on my door. We'll give me 1 second here. Got somebody wanting to come in and work here. 2 seconds. Be right back. Okay, coming back. All right, I'm back. Okay, so if you'll look real quick, the only thing you have for today is if you look at your structure. So once again, here at the top is the PowerPoint. First things I went over right here, okay, that I just went over, okay, the introduction of animals and then just sponges. You did, this was a sport, this was all yesterday, so then if you look down, even though I talked about it, I put a date on it, there's the peripheral coloring guide, okay, it says December 3rd and 4th, due Wednesday the 9th. I have hard copies of this up here if you want, sometime between today and tomorrow because it's a lot of pages, okay? So if you wanted to come up and pick this up, you're here. I've got stacks of them anytime, today or tomorrow. If not, want to print it off and do it yourself, that's fine too. But I have these packets up here for you. So that's the only assignment you have after listening to that painful me of talking about sponges. You're like, okay, Meyer, I appreciate it. Okay, so there's that for you on that particular thing. Okay, so without further ado, let's get back to, where was I? Sponges, Animal Kingdom. Oops. Schoology. Go back to where I was at. All right, so I'll turn the lights on here in a second. Get back to Unit 3. Okay. All right, lights are coming back on. All right, I'm going to turn off the camera. Okay. Boom, boom, boom.